Good morning and good afternoon, everyone. Today we are pleased to have Dr. Bing Yu as the speaker of our webinar. Bing is a Chancellor's Distinguished Professor and Class of 1936 Second Chair, Department of Statistics and of Electrical Engineering and Computer Sciences at UC Berkeley. Bing was formally trained as a statistician, but her research interests and achievements extend well beyond the realm of statistics. Her research focuses on the practice and theory of statistical machine learning, veridical data science, and solving interdisciplinary data problems in neuroscience, genomics, and precision medicine. She and her team have developed algorithms such as iterative random forest, stability-driven non-negative matrix factorization, and adaptive wavelet distillation from deep learning models. Bing is a member of the National Academy of Science and of American Academy of Art and Sciences. She holds an honorary doctorate from the University of Lausanne, and she is also a Guggenheim Fellow. Last year, she is a recipient of the COPS Distinguished Achievement Award and Lectureship. So without further ado, let's welcome Bing, and she's going to tell us about veridical data science.
Thank you, Zhihua, for the kind invitation and very kind introduction, and thank you all for coming. So please feel free to ask questions. I think many of you might have heard early versions of this talk. So the goal of this talk is really try to see whether any of you might be interested in trying out this approach. And so I'll really welcome questions during the talk. Oops, if I can move. Okay. So we're in the era of AI, right? And I think I really like this quote: AI is like nuclear energy, both promising and dangerous, from Bill Gates, which before the whole ChatGPT came about. And most of the advances in AI have a data science component, right? You have robotics, right? That's more mechanical engineering and hardware, but the heart of AI right now, the frontier, is really data-driven. And I want to situate my talk in a new kind of case study that I had previously talked about. So one problem I've been working on with my co-authors is really prostate cancer early detection. Prostate cancer is like breast cancer for men. And about 12% of people in their lifetime in the US develop prostate cancer, men. And right now there's something called PSA, is kind of a screening. And the AUC is only 59%. And if you look at one point, the curve, if you want the 80% sensitivity, the specificity is quite low. And it's invasive. And after it's the false positive, you do biopsy, it's very invasive, and you have many false positives. And the question, can we do better? And another case study is, I think some of you might have heard, is this long-running 5 years joint project with many people, mainly with you and Ashley. The guy is a Thai, and two young people, Chen Wang from UN's lab, and Tiffany Tan, who's at Michigan and going to Notre Dame. So UN is a cardiologist from Stanford. So here is, we try to look at this HCM, hypertrophic cardiomyopathy. Can we find genetic drivers? If we find genetic driver, we might have drugs to mitigate the situation. So this HCM is characterized by the left ventricle wall get thicker, and therefore it's harder for the heart to pump blood, and you have all kinds of bad consequences. And there's a previous work show that there's important genomic components, and cell size seem to be associated with HCM, right? You have big cell sizes, of course, the wall will be bigger. So both are data science problems from my point of view, and there are other parts I'll mention. The first one is more purely data science problem. The second one is actually we did experiments, so we did other studies, a lot more holistic approach. So we go through what's the medical question. We want to have better sensitivity, specificity for prostate cancer detection, what data to use, how to clean it, EDA, modeling, and interpretation and validation. Here we actually have two validation cohorts here on this one. So external validation, not just from the data you have. And every step, we make the judgment call. Humans involved make judgment calls. So two teams will get different results. sometimes. And similarly for the second problem. And the view I hope we all will get more used to is really, we should see modeling, not just the modeling, but really data analysis, a whole pipeline of workflow, data science lifecycle, and people at the center. And we introduce uncertainty, we can also make things more stable. So it's all up to us. And this process view has been written in the statistical literature, start with Box, maybe even earlier, and other people like Carlson, Snell, and Nelder. So statistics really care about uncertainty, but we need trustworthy uncertainty quantification, not just we give some intervals. And if you look at our field, we actually don't validate very much the uncertainty levels, except in simulations. But if the simulations don't capture reality, it's hard to believe that when these intervals work in practice, we'll really do a good job and be trustworthy. So we want to take a step back from the traditional statistical approach, the like sample to sample variability, to really hugely expand that to include human judgment calls. So why veridical data science? So two important reasons I'll provide is that we're actually underestimating uncertainty in a data science life cycle. And we care about uncertainty. I hope everybody here agree. So the two pink elephants, at least we should remove. There are many more smaller ones, but that's for later. It's data cleaning. We have uncertainty we're not taking care of. And for choices of algorithms and models, human make, and we are not taking care of formally. So just give you one piece of evidence for uncertainty administration due to data cleaning. To illustrate the point, I did a study in my class 2 years ago, and students, 3 group students, tried to clean some data to decide whether TBI kids in the ER room who have brain injury, whether they have bleeding. So the gold standard is CT scan, but it has radiation. So you don't want to send too many kids there because the young radiation can cause lifelong hazard. And the 3 teams, each with a UCSL medical doctor, and gave the same raw data, same instructions, and they got 3 different versions. One team got rid of 23% of the data, right? So if you go extreme without any concern about data cleaning set, you can have too optimistic result on your classification algorithm. So we produce more versions. My TA Omar did that, and the Dr. Complex is what I have been working with, and Chen and now Microsoft research with students just, in my group came in to help. And you can see that in terms of sensitivity measurement, if you do bootstrap a different data, same data set, the magnitude of uncertainty is similar to if you choose different cleaning choices. So it's huge. And here's another piece of evidence for uncertainty estimation due to algorithm and model choices, or team effect. There's 73 teams of people. This is a paper from 2 years ago. Social scientists work on the same data, no worry about data cleaning, and try to test the same prominent social science hypothesis about immigration, whether it will reduce public support program and provision of social policies. And the 73 teams get results from the very positive and to the very negative. So this is really something they called a hidden universe uncertainty. And they actually recorded many decisions of this team made, try to analyze where this difference come from. They couldn't reach any conclusion. Just seemed to be kind of reasonable human judgment calls people made. And these people are qualified. They didn't let anybody like Kaigo to work on this. So they evaluate the teams to make sure they are qualified. More recently, when I was in Australia, I talked to some ecologists, and they point me to another paper in Nature last year. Similar conclusion. They have more teams of people work on 2 data sets in ecology, and they got different results. It's the same data, different analysts. Variation in effect sizes due to analytical decision in ecology, evolution, biology. So all of they just point that there's this extra layer of uncertainty we're not taking into account. These people all did traditional statistical uncertainty in each of their approaches. So what are the bad consequences of uncertainty underestimation? There are many. We have too many false discoveries. So the cancer project I'll be telling you next is a result of my giving a talk in Hutchinson Cancer Center 2019, and Ziding Feng, who is a validation center director for the Early Cancer Detection Network of National Cancer Institute, liked the approach because he was facing a real problem. The people who came to his validation center tried to, replicate their cancer biomarkers. They had replication rate is less than 10%. So this is all published papers. So we have too many false discoveries and irreproducible scientific results. And therefore, we have too many unnecessary papers with wasted editorial review efforts, because people just do better taking care of model checking and also stability or robustness, then will really reduce the downstream work for the community. Because when you have this inaccurate or under certainty not accounted for, we're too confident or mislead patients. And a lot of the result, even the clinical decision rules are made without considering data cleaning uncertainty. And we'll have too many failed clinical trials and lab experiments. And eventually, we'll have more public mistrust in science. And then the Congress is not, the public has mistrust, and then going to result eventually reduce funding in scientific research. So things consequences in the long run can be very serious, even in short run from patient's point of view. So I like to take a pause here actually to see from the audience, which the evidence are presented. I would like to hear what you think we should do. I'll tell you what our approach, right? That's the rest of the talk. But I'd like to hear whether people see this as a problem or people don't. So I'm happy to hear whatever comes through your mind. Anybody want to chime in? Yes, somebody raised the hand. Chuck, please.
Just a second, let me unmute.
Somebody raised hand, yeah. Yeah. Chuck, I think you have the floor.
These two studies really aren't such great news, given that we have long-standing replication crises in statistics. Different fields have experienced them at different times. And now we're learning about the medical field experiencing it, and who knows what else.
So you agree that we do have challenges or crises? Thank you. Yeah, so that's how I feel that we have to act, and I can tell you what we try to do. I think that it's our personal responsibility to fix the problem. And we need to go back to what we used to do. Traditionally, we emphasize, when I was a graduate student, emphasize a lot about model checking, model diagnosis. And now, with just so many people engaged, and the methods become relatively easy in software. So before we did a lot of just code out one model. That was enough. But now with software development, that part becomes a lot easier. And we need to worry about more factors than we used to, which is human judgment calls. And our attempt to fix the problem is through this veridical data science, through the predictability, computability, and stability principles. And we need to go by really insist more on reality check, which I don't think we do enough. And then we need to formally address these uncertainty sources, at least from data cleaning and model choices. In our book, we actually formally address these two sources. There are many other, hopefully in the future we'll address, but let's start from somewhere. So for the rest of the talk, I'll do a review of PCS framework. Many of you hopefully already heard. I hope it's a good review for you. And then I'll emphasize on how you might use PCS. So first is that we should stress test every step of data science lifecycle. Even you cannot address every step, but at least address the stability issues in the modeling stage. So we can towards conducting veridical data science projects. But PCS, the way we use it, is not just do stress testing. That's more like assessing instability or trustworthiness. Also, when we find that it's not stable enough, we can actually add stability in and get new methodology development. So both are very important. And I'll finish with the current directions and talk a bit about the book. So PCS, right? This is really built on Leo Breiman's work, two cultures, try to synthesize the two cultures together. Computability is always there from day one. And predictability, when I first started on this path of PCS, I was thinking about use prediction for model checking. It's still the case, but I think it's not enough. So I have expanded P for reality check. And we might want to do subgroup prediction check, not just the average. So it's really P is standing for reality check. Sometimes for transfer learning, you don't have labels. You use domain knowledge to do the P, I would call, which is for reality check. And stability is a huge expansion on sample-to-sample variability. And I chose the term stability because robust statistics has a particular meaning. It's really robustness. And control theory use stability, numerical analysis use stability. So I thought it's a good unifying term. So in one slide, it's a synthesis. If you do many things I say, that's wonderful. I'm just trying to make it a coherent framework and get beginner to use it. It's a unification and expansion. I haven't seen anybody take into account uncertainty from data cleaning formally and really build a platform for further developments and invite the whole community to join me. And it's an evolving field. It's not like set or we figure out everything. We don't. But I think the principles will stay of reality check and robustness and computation. So in a nutshell, it's really try to see the whole process, and then make sure things are stable and make sure things are reality check. After you evaluate the stability, there's an aggregation step, right? Do you take the union of different model result or take an intersection? And that depends on the situation. So I'll get to that later. And the context is very important. All the perturbations supposed to be reasonable, right? If I drop my phone here, sitting in my living room, it shouldn't break. I go to the top of my house, attic or something, drop it there, probably will break. So that's not a reason perturbation for my phone because the iPhone is not supposed to, you cannot sue Apple, say you go to the 5th floor and drop the phone and it broke, right? Nobody's saying it's because it's not reasonable. But if you drop from your pocket to the floor, I think you have a case. So that's why important. The context decides what's reasonable. And you write the documentation about it. So I started falling in love with stability principle like more than 10 years ago. I was given the opportunity to give the 2-key lecture. I was trying to connect sample-to-sample instability of Lasso, which is like discrete thing, and to model perturbation, like what 2-key was considered by in robust statistics. And then there was really a salute of paper at the time about scientific irreversibility. Chuck was mentioning, right? It's just a lot of attention and psychology, p-value, crisis, all of that. And now I really think stability is also prerequisite for interval machine learning or explainable AI. So that's a later thing. And it's a common sense. At the time, I didn't think it was a common sense. It was very technical. I was only worried about data modeling. Now it's about every step of the data science life cycle, even linguistic stability, right? People like me, we learned ML used to be called maximum likelihood. Now the meaning of ML means machine learning. So the meaning of things change. When you have teams of people work together, you want to make sure terminologies mean the same thing for the same team, everyone on the team. So these things I'll run very quickly. It's a unification of many different forms of data perturbation reasonably. You write a document about it, including the unconventional data cleaning. You put in synthetic data or data augmentation in AI and different variance consideration in causal inference, invariance consideration, and privacy and adversarial text. So this is an attempt to unify many things, and we can see similarity and discuss their differences, and really build a knowledge base in a hierarchical manner. For my own memory, I cannot remember a lot of flight things. I need to organize them and through basic principles. And I started with robust statistics, and now we have choice of models. So I'm really advocating for consider multiple models for any particular data science problem, unless the domain, the context say this type of structure model is the right thing to do. But you might want to expand that to see whether the models you think you like linear models, can be augmented and improved by, say, random forest type of models. A quick question in practice I'll get to is like, how do you choose the perturbations? Because if you do every possible way, you never go home. So that's where judgment call comes in again. You know, where is the pain point, where you're not trusting most, and you want to poke that to make sure things are stable. And depends on the resource, human resource and computing resource, you will do only certain number of them, and you write a document about it. So this is very much philosophy related to Gell-Mann. Loken has been more in the Bayesian context about forking. So you can weigh, or you can take a few paths. My recommendation is that you should at least have 2 clean data versions and just run through the whole pipeline to see where you make a difference. If you do, then you have to go back and really think about what's the right way. Or you might just say that I just have to add uncertainty and documentation. So models are mental constructs that only connect to reality because you make good evidence argument, quantitative and qualitative, and you document it. Otherwise, applying a model to data doesn't mean the model captured the right reality. It can completely distort reality too, because the structure of your model is not capturing reality. And we have a template came out from a collaboration with the MD Anderson people that you can take and then revise for your own purpose. So take a step back. PCS is really a philosophy. It's a conceptual, practical philosophy that standing on basic principle, we might even add other principles later. So I really see this as evolution. It's not a set thing, and invite everyone to come together. The goal is for veridical data science, truthful, trustworthiness, and trustworthy quantification. It's a system approach. I want to see the different steps together, and with expanded uncertainty quantification, which is closer to reality. So veridical in the title means truthful. In the beginning, when Tianzhen from Columbia recommended to me, which I love taking, it's really about try to say we want to achieve truthful conclusion. But now I think about it, it really means we want the process, what we teach in the book, to be truthful to or close to what people do in practice. So now it has multiple meanings, and we do documentation seriously. So now let me go to the first case study, which is prostate cancer. Right, so we brought in, because we had a successful case study with about pancreatic cancer with metabolites with MD Anderson group, many people engaged. And then I was invited to give a keynote to the PI meeting for EDRN, early detection of cancer network detection for National Cancer Research last September. And that was 2023, like spring, a year ago. I asked Ziding, I said, I want to go in with 2 case studies, not just one. So he introduced me to Arun Shenayan, the doctor on the side, and then we work with Yuping Zhang, the data analyst on his team at University of Michigan. And Tiffany, I already mentioned, Anna is a young professor, my former postdoc at UC Irvine. So they already had this archive paper. So we went in as what we call stress test, redo the analysis, and to assess uncertainty from data cleaning or process. And they were very gracious to let us do that. So this is, I'll have to go a little fast with interest of time. Then there have clinical variables, I won't mention too much, more traditional. And they were looking at gene expressions, and they end up with 54 gene expressions. They were selected based on predefined normalization criteria and differential expression, so kind of data quality control. Then they have something called CT. CT is a cycle threshold. When you do RNA-seq to measure gene expression, you want to know how many cycles you want to have amplification, and that's a quality control, so they do thresholding on that. So that's an important threshold. And they also have clinical outcome. So for them, the clinical significant prostate cancer is a grade group 2 or higher, so the most serious ones. So that's the prostate cancer definition here. And they have this EDRN threshold, validation. They end up really improve the state of the art, right? So the sample size 775 and clinical variables, the typical age, family history, and the things they have been using traditionally. So now this is more modern with genomics and gene expression. And so they have something called MPS2 because they have MPS before, and they use logistic elastic net, okay? So here's some validation data, right? So they really improved the state of the art. The validation data is about 859 individuals, average age 62 years old, and MP2 plus is the gene expression plus the clinical variable. Plus means plus the clinical variable. And they end up with 17 genes, use elastic net. So you can see that PSA is what's being used. They improve by 21%. And PCPC is only on clinical variables, something also used. They improve by 15%, and their own method, MPS, based on 2 gene expressions and clinical, they improve by 7%. Okay, so this is all AUC, and you can cut at different places. So we talked to Yuping and really try to stress test, right? We want to reproduce and assess uncertainty from data cleaning. So the important thing, this is also human judgment call because you cannot try all possible ways to clean the data. You want reasonable ones. So the CT value, we probe that, and you can have different ways screening up poor quality samples. And we decide to go for 4 other possibilities, right? This is the judgment call. So if you take the PCS framing, you don't consider only 1 models. We considered a bunch of more lasso-rich linear models, and also nonlinear model, random forest and tree-based method. Later we have other things. And then it turns out that so the stress test recipe is identify human judgment calls, decide which one to do reasonable permutation. We decided to perturb the CT threshold because we thought that was the importance. You can see we just a meta level human judgment calls. And we couldn't do 100 of them. And we made judgment, we did 4 of them. And then for each perturbation, we evaluate performance metrics. So this is the reality check part. We only care about models for aggregation if they do well. We don't want to aggregate a bad model with a good model. So we do prediction check. And then we did stable ranking because we now have 6 different methods. Each has some importance measure. They rank the genes differently. And we want, if the gene is important, among all the good models, actually they're all pretty good for this case, then we want the gene appear in most of the models. So this is kind of thinking is really a bit like pseudo causal inference. The different structures means different pseudo causal environment, and you want the invariance because you kind of get the pseudo causality in a way. And you can look at a subgroup accuracies too. So they came out really well with the pre-processing. The accuracy change, if we do the 4 different ones, about 1 to 2%. And they have 7 to 20% improvement. So they are completely safe, right? You can see that that's the different changes, right? There are certain thresholds and the accuracies. So this is good news for them. And logistic L2 actually has relatively low AUCs, right? That's a lower group with 4 different. So each block has 4 different ways of processing, and these are the scores. And you can see listed, and that's pretty high up. But random forest, I'll mention a bit. Random forest plus is something my group developed, building on random forest. And then random forest not as well but they all kind of pretty much better than logistic L2. This is using 54 genes. And then we look at the stability of rankings. 3 genes always top in all 17 genes. So that's why they, the top 17 genes and top 3 genes always in the top 17. Doesn't matter which method you have. And I'll have only one slide about new method called RF+. We have a paper called MDI+. It's really try to have more inclusive way to look at the model like feature importance. So we combine the trace nonlinear feature extraction with the linear to have some feature importance, taking into account both linear and nonlinear effect of a feature, nonlinear only through the trace. And we have MDI+, and as a result, also have random forest plus, which is basic generally modeled on this new blocks of features. So take a look if you're interested. It's on my website. And so we use that. And it's very much PCA driven or PCA guided, PCS guided. So for each feature, we look at now mean feature importance ranking across all pre-check models. Logistic 1, we deleted logistic L2 because the performance is worse. And so we have actually 4 methods. We passed the prediction check, and we got the stable ranking. And we use the top K rank features plus the clinical variables to make predictions. And we couldn't improve the performance very much, but we really improved interpretability, sparsity, and cost. So this is just some stability genes. You can see the top 17. We start from 0, that's why we end up with 16. Right? We have 4 different thresholds for this particular method. So in the end, we end up choosing the top 7 or top 6 use stable ranking. And then we actually go back to L2 panelized logistic for the prediction. So you can see that the method, even by itself with 17 genes, didn't work so well. After you help reduce to more like important genes, much smaller, and that turned out to be a good method. So it also says that you should detach a discrete problem, which is ranking, from a continuous problem, which is how you're going to use it for. I think it's more advantage in many problems to get the ranking first, the right ones, and then find a method, then kind of do them together. That's what LaSue does. So I think for the ranking, you need to rely on multiple methods. It's more trustworthy and I think has more chance to be causal. So now you already saw the panel on the left, right? That's the 17 genes plus clinical. And on the right, with the stable ranking, we're able to go to 6 or 7 genes. And you can see that we're very comparable. Tiny bit worse, maybe 1% on the first validation cohort. It's about 659 individuals. We actually have another validation cohort, which is from University of Michigan. It's a little smaller, but still about 500. So we compared the two, and then it really become, remember for the EDRN cohort, the best one from our best with 7 genes was like 1%. But here we basically, to the second decimal place, we're on par with that, right? So this is really says the plus means plus the clinicals. And we reduced the cost and interpretation from 17. That's like cut of like almost 6% of genes. So now we're writing it up, and hopefully this can go into clinical use because now it's much less genes you have to collect. Instead of 17, you only need 6 or 7. So the takeaway is that whatever our Rules group did really passed our PCS stress test for the clinical, for data cleaning with flying colors. And then guided PCS were able to help improve their interpretability and sparsity of their method and reduce cost. And 2 external validations, that this is just last couple of weeks, confirmed the result. We have two actually external studies, not just one. Any questions? So that's a more typical data science project, right? It's mostly statistical. And with the, of course, data collection part. Our involvement was not on data collection, was really the statistical part or data science part. So the second project I spoke at the JSM is more extensive. We engage in problem formulation or reformulation. And the second part is the recommendation. And we did annotated database search. And we work with them for the gene silence experiments and analysis because our feedback, the redesigned experiment, and then the mechanism interpretations we engaged, they led to a lot more complete, I would say, engagement with the process. Basically, we're hoping that the gene interaction we find of genes eventually leads some drug treatment, right? So this is a lot more extensive than the first project. It was like, the data was there, problem was already formulated. We did the statistical part. But I think we should engage more and more in a more broader scope with these projects, because I think people do welcome our engagement, even for the experimental part. So this one, I'll go quicker because I think you heard it at JSM. So we had to, the labels didn't work. We have to go back to the MRI, get new phenotype, which is the volume of the left ventricle heart chamber wall. And then we couldn't pass P, the prediction, because we don't know it's a regression problem. We don't know what's the noise level. So we binarize it to make it a balanced classification problem and also enhance the signal when the people with very big heart, left ventricle wall and less, they are cleaner signal. And we tried stability right there, right? So stability principle is right there. Is no feeding, but want to make sure the thresholding doesn't change the result. And we're able to beat the benchmark, but only 55 accuracy. I've never worked on real problem with so low accuracy. In IT, it's always higher. But at least we know it beat the 50%, which with ACM labels, we couldn't. So there was a lot of faith, annotated database, and work with the best experts we have to have the confidence to do experiments. So this is the UK Biobank data, SNP. And we have to use data dimension reduction. If you heard of my earlier talk, we have this iterative random forest, but usually we can only take like a few thousands dimensional features for two reasons. One, the computation. The other is that we're looking for nonlinear interaction. We have too many, you basically have too over parametric model, you probably have too much noise. So we need to do dimensionality reduction. So here we have two use marginal GWAS to pre-select some SNPs. So here we did two leading software, both LMM and Plink. We didn't do the intersection. We did a union because this stage is very early on. We need to keep all options open. They overlap about 50%, and other 50% they are different. So put them together. So aggregation for stability is not always intersection. For the later stage, you might want to do intersection. In the beginning, you need to think. Sometimes you might want to do intersection, but for this, we decided we're going to do the union. So with the pre-selection, with the binarized problem, we tried 3 different thresholds. The stability right there. You can see stability is everywhere, even the reality check, of course, really rely on the data being good, which we didn't deal with directly. And then we did iterative random forest on the SNP data. Actually, it was not stable at the SNP, too many SNPs. So we aggregated the interactions at the gene level. You can map each SNP into a gene region. So we collected that and get stability. So every step we have to add extra stability consideration on top of what we had already, like iterative random forest. Iterative random forest for low signals is not enough. So we call low surf because for low signal. And then we did days of annotated database, talk to them. This is recommended that when you work with your expert collaborators, don't give them just what you find. Give them 3 lists. The ones you like, the middle range ones, and random one to see whether they really can pick them apart. We did many database, annotated database search, so I'll be brave, and then we did experiments, which we also may contribute. They redid the design because there were some big cells clumped together, and we're looking at cell size can completely skew the result. So in summary, 5 sets of experiments, 4 found strong causal evidence. And we took another 6 or 8 months to bring in other data set to come up with a mechanistic interpretation. And we just sent back the revision to Nature Cardiovascular Research, and the reference was great, asked many good questions. So our response was 38 pages. Because this is really serious stuff. The genes we find, the interactions, it's very, very hard to find in human diseases, right? Mostly it's marginal. And I think a lot of this epithesis not being able to find, it's because the Wignall's relative is complex. And if you want the p-value to be less than 5%, it won't pass it. But this complete approach uses ranking as recommendation and then do experiments. It's the way to go for epithesis discoveries. That's my what I believe. Because if we only use the data without experiments, we won't pass any of them. You know, the prediction accuracy is 55%. You don't have really high p-value. But as ranking is a lot more stable. So the rankings still give information for us to do experiments. So again, shout out to my main collaborators on this project. So to summarize, the pipeline is 3-way split, right? Training, validation, test. For the other MD Anderson split, the book, we have 3 different splits: random splits, group-based splits, and time-based split. So the book, we really talk about what's a good test set. You want to imagine the future situation. You want to find the best proxy, the data you have to mimic the future situation. For the MD Anderson data for pancreatic cancer, the data come from different hospitals and centers. And the metabolite will be used for different hospitals. So there you have to do group split. Here, the data is relative from one hospital or something. So we didn't do the groups, but we have multiple groups, right? In sometimes we did that way. So think about what's the best proxy to your future usage. And that's the best we should set up the split to mimic as much as possible. Don't always go for random split. And then we did more domain-inspired reductions, and we did stability, right? We used 2. We did a binarization, and then we also did pre-selection. We have very big signal. So throughout, we have to struggle with get some signal out of a very complex situation. Here, we actually also tried SVM, and we did a stability check, and we have validation through lab experiments. And there's another part which is mainly led by Ewan's lab about getting more data, more interpretation about the gene, the epistasis we find. So in case some of you, I hope you do, some of you might be ready to try PCS. So let me just summarize what I think PCS can help your result. We got so much mileage out in my group's research, traditional research, using the PCS. We always take the stability angle and the model check angle and documentation for every project. And that's how I teach in my class, 215A in Berkeley. So it's really common sense, right? We need to do stability robustness check. And the aggregation is a little, you need to be thinking about, do you do the union, you do the intersection, or something else, right? But when you first expose the instability, you can just do the ensemble. That's one way to aggregation too, which we haven't done in the ranking. We did the aggregation there. So we can improve through new pipelines and get better results, right? You see in the prostate cancer, we didn't improve the accuracy, but really reduce the number of genes. So that's new methods. We stress tested the prostate cancer pipeline. And then for the cardiology project, we had more reliable factor to recommend to control for downstream experiments. So I believe this is empirical. the PCS framework give you more internal validation so that you can have higher yields for your experiments. So we reduce false positive. You can never eliminate completely, but we reduce with the current approach. And we're really now working on more realistic uncertainty quantification. The goal is reproducible scientific results and trustworthy domain knowledge. So 2 concrete suggestions for your data cleaning step. Document it. Just keep 2 versions, say 2 thresholds. You cut age at 60 or 65. Either you're very sure, right about it. This is the right cut. Here's the references, why this is a good idea. That's completely fine. But if you don't, often we don't. I have never seen any of my data cleaning projects that I know just one way to do it. There's other reasonable choices, so I tried 2. And you just rerun the analysis pipeline to see whether qualitative your conclusion hold up. If they do, it's not waterproof, at least you have some confidence. If they don't, then you really have to decide to either take this, say there's no qualitative conclusion that holds up, or argue one way of cleaning the data somehow is the right way, and you have to make a case in your documentation. The second suggestion is in the modeling phase. So you say, I actually don't do data cleaning yet. Hope you do in the future, because it's always good to clean your own data if you can. It's the modeling stage we all do. We actually try different models, but we have the habit of reporting only one model. So my recommendation of PCS is like, actually you report on multiple good models. And just say they're actually not separable. They're all very good. After, say, you take the top 10%, that's also, you have to argue why that's a good idea. And then you aggregate through the stability principle. You can say union or intersection or ensemble. That's all legitimate. And then you document everything. I think this is something I would think most, or should every statistical group are equipped to do. The question is whether you want to do it. I hope I show you enough evidence in my own traditional research through the stability principle. There's a lot to be gained. And it's not that difficult if you buy into this common sense principle of stability. And now the scientists actually are telling us, we want to worry about this. I don't think it's really up to us to say that we don't want to worry about this. The multiple approaches, right? The 2 papers from domain fields, social science and ecology, that people writing papers tell us that, how do we deal with this? So if you want to be a little more advanced, you should also go back to reformulation. Maybe you have different data, maybe it will be better. And for the inference, we have a little bit in the book, and we actually have our first paper on PCS inference published for epistasis polygenic phenotype discovery. We changed features model from logistic regression to actually tree-based method, and we have more robust p-value calculation. We didn't worry about data cleaning in that paper. It took a while. It finally appeared in +1. So this is still very much active research in my group, and hope many of you will join me to make it better. And I mentioned iterative random forest, but I didn't have time. It's more data-driven. We actually developed many new methods using adding stability to existing models, right? We added stability to random forest. How you add it, context dependent. And we usually develop methods in my group most of the time, I hope, through problem solving. And we use stability to choose the number of components for non-negative matrix factorization. Statistics is use stability-driven PCS, and we have to be creative to decide what's P in causal inference, randomized trials, because there's no labels. We have to do aggregation, do calibration as model checking, or P. And stable subgroup discovery at the Statistical, and we have another paper for drug discovery called StatDrip. We have been working on software. There's one Python package called the vFlow. Go to my website to make the stability analysis streamlined, building on Berkeley CS software. It's a wrapper on top of MLflow and Ray to try to do a parallel computation. And I didn't have time to talk. A big part of C in PCS is actually data-driven simulation. So we have a simulation in R package, especially for linear model, to make different models. You should fit different models, good model to your data, and simulate to see whether your method works. And we make that easy. And we're also finishing a paper, just put it, submitted on guidelines, how to do good data-driven simulation. So this is a package just appeared, both is accepted by JOS, a journal open source software. So people actually look at your codes and all of that. So check it out from my website. I have an upper corner, there's a code link. Documentation. We developed this with MD Anderson, and you can just take it and put in your problem, especially for beginning student. It's really helpful, help people to organize things for you. And I think it would be good for Statistical Journal to have consider something like this as a supplement for all our journal submissions. So the referee job will become a lot easier because you basically organize all your thinking in a very kind of boring, but very systematic way for referee to check how, what's the problem. For paper, you can write more freestyle, but with this documentation, we can make it more standardized and just make referee job much easier. And we all benefit because we all referee other people's papers. To summarize, so PCS is a philosophy, basic principles, and you can improvise. There's a lot of creativity involved to make it work. But we have done many good case studies. You can take a look, and we got a lot of mileage out in the traditional research metric. So it's internal validity check. And we did recommendation for internal causality validation. Domain knowledge is always very important. Without saying everything's in context, that's the recommendation. And you can evaluate other people's work. And decision we did too for the prostate cancer and also for ER decision rule. And you can also develop traditional, like new algorithm in the traditional sense. When you know something is not stable. So RRF, iterative random forest, was because people have the idea 2 genes on the same path of the tree might interact, but it's just so unstable. So we started with a good prediction algorithm, iterative random forest, and then we added stability. And that's become nonlinear model selection, has been like a workhorse in my group. For the cardiology problem, we do have to add a lot of bells and whistles, but it's just all stability driven for low signal problems. And many people actually have extended the framework to veridical data science, called spatial statistics, veridical spatial data science, veridical network analysis, and reinforcement learning. And some people start working a little bit on theory. So a lot of papers on my website, and I won't go through, and there's many. So now I actually organize my papers into different subcategories. So you click the paper, before it's just all long list. Now you can see the subcategories, and you can go to the PCS papers more easily. I hope we'll at least remove the 2 pink elephants in the near future, with many of you joining me. We have a workshop. Its registration is open now. If you go to my website, you can have the link. It's the first Berkeley Stanford joint workshop on veridical data science happening on May 31st. And if you register, you can also submit to be selected for a lightning talk. And all the speakers are there, and it's a great lineup. To quote my collaborator from MD Anderson, I want to ask him why he ended up liking PCS. He said, at the dawn of the AI era, it's extremely valuable to have a uniform language and framework to talk about stress tests in the analysis pipeline. I think we do more than stress test, but that's a very important role that a PCS intends to play. So let me switch for the last 2 minutes for education. Some of you might have the fire chat, so you have seen it. So I want to remind ourselves that our classes, our education has a purpose. I would like to hope you all agree that we want our students to be able to produce computational reproducible and scientific reproducible data result in context. It's both reproducibility and replicability. Some people call scientific reproducibility replicability. And my education philosophy with the very, very fast moving world, especially in AI, it's really the growth model. We want to help the student learn how to learn so they don't need us. So they can pick up new things on their own. And to do that, technical skill is not enough. We need a collaboration culture, interpersonal skills. I learned so much from my friends and collaborators. and my students, because you cannot keep up on everything. And I still find the best source is the people I trust. So everybody helps screen out the noisy papers and get to the quality papers and the software and things like that. So you need to have interpersonal skills to do that. I believe if you take a critical view of our curriculum, I feel like our statistical curriculum need to talk about data science lifecycle. Go back to George Box, talk about investigation process. And we need to emphasize more on computational reproducibility. We just had a faculty meeting in Berkeley yesterday. Many of us, quite a few, had an ad hoc committee, want to have some computational competency and the effort led by Chris Peshaw, my colleague. Want to make sure that we might want to do a workshop before everybody comes to have basic version control, unit test, and just we can put it on the same page. Maybe we should all learn Python as well. I don't think R is enough anymore. And we should also have skill to do deep learning. My view is that deep learning becoming for scientists like linear models. We need to know deep learning and reinforcement learning. And scientific reproducibility really relies on critical thinking. It's pretty vague. It's not quantity. You cannot solve scientific reproducibility by X and Y because you make assumptions. And how do you know the science makes sense? You need narratives, look at the qualitative result and quantitative result by other people. And we also need to make optimization part of our regular curriculum. And leadership and team culture, ethical training. We need to really have expanded scope of our education so that people are ready to learn on their own. So the book is out, free online copy. I cannot share the PDF version because MIT Press still wants to make some money. And the hard copy will come out October 15. You can pre-order already. It's with my former student, Rebecca Barter, now professor at Utah University. And it's really very much guided by the PCS framework and take care of the whole data science life cycle, including a whole chapter on data cleaning. And we have 3 kinds of exercise too. So the website vdsbook.com, and then you can pre-order on Amazon or on MIT Press. So people say, how does it differ from traditional books? So we organize through data science as in practice. And we move away from the framing of true model. In practice, most of the time, anything in my life as a data science, I have never seen a model I can take it as close enough to call that a true model. I don't say that it's never the case, right? But in my work, I have never seen a case I can say, unless even when you do sampling to randomization, you're going to have data cleaning issues. So it's never exactly as a true model or even a structure. So you always have to struggle with the form of the model, the structure of the model. And that's why you want to consider multiple of them. And we fill the gap between domain problem to symbols, right? You cannot do more math to fill that gap. And we use 3 principles. And we teach the traditional techniques and supervised learning, data visualization through the PCS overlay. So always do a stability analysis. When you do visualization, which color you use makes a difference. So you don't want your conclusion impression is dictated by the color. Oh, violin plot versus a box plot. I always like box plot, right? It gives you different impressions. So right there, you have a stability issue. We address 3 examples, case study in the book. And we have 3 kinds of exercises, true and false, conceptual, math, and data analysis and coding. We have codes shared too. If you go to end of every section in the book, you will see the exercise session with links. And we formally address 2 new sources uncertainty, at least consider. And we end up with some perturbation intervals. So it's intended for upper division and beginning stat and machine learning courses as a primary or supplement textbook. I usually teach it with the book of David Freeman. So I use also traditional, but we try to not have too much overlap. And anybody who enter data science, it's really, I think people tell me that it's very accessible for people who don't have data science and statistics background. So it's really a multi-purpose book. Thank you all.